Yeah, thank you for the introduction. As you all know, my name is Peggy, and I have, during my master thesis, done this research on melanoma. It has a bit of bioinformatics, can be a bit intimidating at 8:30 p.m. to suddenly be hit with bioinformatics all of a sudden, but I'll try to make it pretty simple and easy to understand. So, why melanoma? Melanoma is a very aggressive form of skin cancer that we all see, unfortunately, in European and Western countries a lot. And it has a high mutational load. That means that the tumor cells mutate quite easily all the time. And that makes it pretty hard to treat. Now, we have done novel therapies, specifically in immune therapies, which help our immune systems detect and eliminate these cancers. But there is still a lot to do because 50% of the patients that go through immunotherapy end up not responding to it. And that makes us come in to see why that happens, what is happening in the tumor microenvironment, and how can we tackle that. Now, to do it, we did something pretty new. We used independent component analysis. That's ICA. ICA is a statistical algorithm that allows you to detect signals that you otherwise might miss. What does that mean? For example, in this room, if everyone is talking and you're talking to a specific person, you're hearing everybody else, but you're also focusing on the person that is talking to you. You don't get distracted by whatever else is happening. That's your brain doing independent component analysis. It is receiving all the signals, but is focusing on the signal that is right in front of you. In gene transcription, so we look at the microarray data, and we see all these signals from all these genes that are active from the tissue or cell cycle or cell culture or whatever that we have. And we often, if we look at it with normal statistical data that we often use, we see the signals that we're supposed to see. But are there signals that might be weak and you just might miss? ICA is often not used in biology or medical sciences. It's mostly an algorithm that is used in image recognition and signal recognition in physics, mathematics, and other forms of science. So what we came and we decided to see, let's use ICA on gene transcription data. Let's see what we find. And to do that, we looked at the transcription data of 529 melanoma patients. They were biopsied, and the gene was analyzed and uploaded online, and we downloaded those, checked for quality, and ended up with 529 patients that met the criteria. And we looked at what's happening there using ICA. Now, why CoA, vitamin B5, PANC 1, 2, 3, 4, what is happening there? CoA is actually a molecule that is fairly important in biology. It is the basis of life. Without CoA, you're not going to survive. This is something that we all knew for quite some time, but we didn't really look at whether CoA or its pathway or the enzymes that are involved in it would have an influence on pathology, for example. But back in 2020, the paper came out that claimed that CoA and its pathways can actually influence how T cells interact with cancer cells. That if you give them CoA, for example, they recognize cancer cells a lot better. So what we did during my bachelor's while studying medicine, we looked at is this true? Does the CoA pathway actually do that? And we came to see that, yeah, it does. And for my master's thesis, we decided to go bigger using bioinformatics. And we decided to see, instead of the CoA pathway, whether the enzymes would play a better role, because that would be easier to look at. Now, as you can see here, you see that the enzymes that are involved in the CoA pathway, you see PANC 1, 2, 3, PPCS, PPCD, and CoAC, but you don't see PANC 4. That's because we thought until now that PANC 4 was just inactive, something that was a result of evolution and was just there. But research shows that PANC 4 is actually acting as a phosphatase. And what happens is that you see PANC 4 interacts with the enzyme PPCS, so not directly with the CoA, but with this enzyme. If PANC 4 gets active, PPCS gets inactive, and you don't get the downstream here, so you get no CoA. It's quite interesting. Can we see that in melanoma? So we looked at the data. How we did it was that we got the samples, we ran the independent component analysis, and what we did was that we looked at what we caught. So a component independent analysis gives you independent components. Those are the signals that captured, and you look what genes were captured in that signal. Surprisingly, we caught PANG4 in IC38. And what we looked at here is what we see that IC38 catches PANG4, and not only it catches PANG4, but if you run a correlation analysis against from IC38 to all the other ICs that we've caught, it's negatively correlated with IC101, and that is PPCS. So what we captured was actually positive control. This showed us that what we are getting is actual data. We're not just getting stuff. And then we ran a gene ontology analysis to see the biological mechanisms that are involved in this IC38. So what is PANG4 actually involved in? What is it doing? We saw that it is involved in RNA processing and transport, cell cycle and division machinery, and immune activation and antigen presentation. That is quite interesting. Now, RNA processing and transport, you expect that. That's just cells doing their cell things. But cell cycle and division machinery, that is quite important to something like melanoma. Also, so is immune activation and antigen presentation. So it could be that PANG4 is actually helping melanoma cells thrive, or it could be helping them with immune evasion. And if we look at its negative correlation with PPCS, it really could be that, for example, that lack of CoA could lead to better outcomes for melanoma, not for the patients. So to summarize, we ran ICA, something that is involved in other forms of science, in biology, and we captured data that actually made sense. Our null hypothesis was that we would see PANG4 or any other CoA component randomly generated in data, and that would make no sense. But we actually saw that the data was showing us a pattern that coalesced around the cellular activation, division, and antigen presentation. For future, what we plan to do is to further data mine to see whether other components can be captured, and also to see whether we can capture the same results in vitro and in vivo. Thank you for your time.
Thank you.